Hi friends, today we are going to discuss about bilirubin formation, uptake of bilirubin, conjugation of bilirubin, and excretion of bilirubin. So let's begin. Let's go through that how the bilirubin is formed. We know bilirubin is a greenish yellowish compound which is formed due to the breakdown of RBC cells. When the RBC cells become old, they are destroyed by the macrophages in the spleen, and after that bilirubin is formed, iron is formed, and amino acids from the globulin they are formed. Iron and globulin they are used while bilirubin is excreted in bile. So for example, we have RBC cells. When the RBC cells become old, for example RBC cells, they complete their lifespan of 120 days. Then these RBC cells go, when they circulate in the vessel, in the blood vessel, and reach to the spleen, there present macrophages which destroy the RBCs and produce heme and globulin. Globulin and heme. From heme we get iron and bilirubin. We will discuss all these steps in detail, little bit detail, and with a presentation. For example, this is your RBC cell. If you look the RBC cell, they have a specific type of protein called spectrin. In these spectrin proteins, they are responsible for the flexibility of RBC cells. RBCs. For example, this is the vessel, capillary. They can ooze out. They can flexible, and they can ooze out into the interstitium, into the cell. Okay, they will go out and in like this. They pass through the capillaries. So once they lose their spectrin protein after 120 days, these RBCs into the spleen. Okay, into the spleen. They are stuck into the endothelial cell, a very fine network of capillaries. They are called endothelial, reticuloendotheliosites, and cord of Bilroth. We can also call like this. Okay. So the RBC cells, they are stuck here into the capillary network. When they are stuck here, macrophages present in the RBCs, they come and engulf these old RBCs. So for example, there is your macrophage, and this is your RBC. So these RBCs are engulfed by macrophage like this. Okay. No diagram I make directly that the RBC cells, they engulf the macrophage. RBC cells engulf, and they are broken down inside the macrophage. And this is extravascular because it is not inside the vessel. So here what happened, into the macrophage, the RBCs, they broken down into heme and globulin protein. We know that heme is made up of protoporphyrin ring 4 pyrrole rings, they combine and they form protoporphyrin ring. When iron is added in the center, they are called heme, and this part is globulin, which detach here. Okay, so here this is heme. It will divide it into iron, and the destroyed or damaged protoporphyrin is called biliverdin. This biliverdin then convert into bilirubin. So how the reactions occur? Let's see. For example, this is heme, convert into biliverdin first, then this biliverdin convert to bilirubin. Here the enzymes which work, they are heme oxygenase here, heme oxygenase enzyme, addition of oxygen, removal of iron, and removal of carbon monoxide, formation of biliverdin. And here the enzyme which work, biliverdin reductase. The biliverdin reductase work here and convert the biliverdin into bilirubin. This reaction, the first reaction, is called as oxidation reaction. Oxidation of heme or oxidation reaction. And number second, a reduction reaction. And then the reduction reaction or reduction of biliverdin. So in this situation, in this case, bilirubin is formed from the RBC breakdown. So there are certain complications. For example, when the bilirubin level increase in the blood, for example, when severe hemolysis, for example, autoimmune hemolytic anemia, or anemia or hemolysis due to any other enzyme defects. For example, in glucose 6-phosphate dehydrogenase deficiency also leads to hemolytic anemia. So all these situations, okay, in all these cases, excessive heme is produced, or excessive breakdown of RBCs produce excessive bilirubin. The bilirubin in the blood very high. And which type of bilirubin in the blood will high? That is unconjugated bilirubin. We will discuss that what is conjugated and what is unconjugated bilirubin into the blood. We will discuss later. So this condition is called only one kind, okay, that is called isolated hyperbilirubinemia. Why isolated? Because only unconjugated bilirubin, hyper in excess amount, bilirubin, emia in blood. So this is about the production of our bilirubin. So next we will discuss about the uptake of bilirubin.